Hello, and welcome to the Chapter 3 lecture for Crowley's An Introduction to Human Disease. This chapter is all about genetics and cell division. So we know that the nucleus of the cell is where the DNA is stored, and that the DNA in the nucleus is coiled up into these tight bundles that we call chromosomes. So all of the DNA in the cell, in the nucleus, and also the DNA in the mitochondria counts here too, but the collection of all the DNA in the cell is referred to as the genome. All right, so the genome is the collection of all of the chromosomes. Chromosomes are just large pieces of DNA. Every cell in the body has the same genome, the same chromosomes, the same collection of genes. All right, a gene is a segment or a sequence of DNA nucleotides. Nucleotides are sort of the letters of the DNA, the GCATs, all right, that code for something, usually a protein. So even though our cells all have the same genes and chromosomes and genome, they will have different phenotypes or different cell types because they express or make different proteins. So that's how we can have liver cells and muscle cells and skin cells. Even though they all have the same DNA, they make different proteins, which causes them to differentiate into different cell types. So we're going to talk about this process at every step in this chapter. The terminology here is important. You need to make sure you understand the difference between these different terms. And my analogy is that a genome is like a book. This is supposed to be a book, okay, with lots of pages. All right, it's the total collection, the whole story, all right? The chromosomes are like the chapters in the book. They are chunks, large chunks, right? So large chunks of DNA in the case of a chromosome. And the genes are like the words on the page. So they're individual units, they're short, and they each have their own meaning, right? Each gene encodes a different protein. And words are made up of letters. And the nucleotides are also, in a way, they represent letters. A, T, G's, and C's, the genetic code. All right, so the nucleotides are the letters. The genes are the words. They each code for something with meaning. In this case, each gene codes for a protein. The chromosomes are chapters. They are just collections of words or collections of genes. And the genome is the whole book. It's the whole collection of all the words and chapters. All right, so different organisms have different numbers of chromosomes, and they are usually in paired sets. So human reproductive cells, which we call gametes, gametes are the reproductive cells. The two types of gametes we have are ovum, or ova for plural, and sperm. All right, each of these contain 23 chromosomes. 23 chromosomes of different sizes, and they're kind of ordered by size here. And since they only have one set of 23 chromosomes, we say that they are haploid. But when fertilization happens, when the ovum and sperm fuse together to form the zygote, all right, the zygote has two sets of 23, or it has 23 pairs of chromosomes, one from the ovum and one from the sperm, so that's why they're color-coded here. So we say that a zygote has a total of 46 chromosomes, but it is diploid because that's really two sets of 23, the 46. So di is a prefix, sorry, 23. Di means two, so two sets, pairs. So the zygote has 46 chromosomes and is diploid. All of the cells of the embryo and the developing fetus and the adult human are also diploid, with the exception of the gametes. So sperm and eggs are the only haploid cells in the human body. All the other cells are diploid. They contain 2 sets of chromosomes, 1 set of 23 from the ovum, 1 set of 23 from the sperm. If we were to take a cell in the body and smash it open and stain and organize all of the chromosomes, pull out all the chromosomes and stain them, we could take a picture of all of them. We could line up these pairs and we can number them. And so this picture of the set of chromosomes is called a karyotype. And the karyotype is a really nice way to visualize all of the chromosomes in a human cell. They're typically arranged and organized by size. These different chromosomes, these matching chromosome sets, we call them homologous chromosomes. Homo is a prefix that means same. So homologous chromosomes they are 2 of the same pair. All right, so they're part of this matching set. So these are homologous chromosome 13. So the first 22 pairs of chromosomes in the human genome are called the autosomal chromosomes. And the last pair, set number 23, we don't call them 23, we call them the X and Y element, and we refer to that pair as the sex chromosomes. And that's because many of the genes required for sexual development are found on these chromosomes, and they are the one pair where they are not always identical. So you can have an X and an X, or you can have an X and a Y. So that leads us to talking about sex determination from these sex chromosomes. But first we have to really define what is male and female. So phenotypically, based on the sort of presentation, the biology, the structure, all right, phenotypically a female, we would say, is someone who has female reproductive parts. So a vagina, a uterus, ovaries. I can't write that, but vagina, uterus, and ovary. Here, we'll try to draw them here, okay? So phenotypically, we would say they have a vagina, uterus, and ovaries, and genotypically, we would say that they have 2 X chromosomes. For males, we would say a male is one that has a phenotype of a penis and testes, all right, the male reproductive organs, and genotypically, we would say that it is someone who has an X and a Y chromosome. Okay, so it's worth noting that these definitions of sex are overly simplified. So in reality, there are genotypes and phenotypes that don't fit those definitions of what we consider male and female. For example, there are people with the male genotype, they have an X and a Y chromosome, but they develop phenotypically more like females. They end up actually kind of in between. So they have a vagina externally, but they have testes internally. So they don't really meet the phenotypic definition of a male or a female. There's also people who there's a variety of different polyploid genotypes where people have multiple X's or multiple Y's and various phenotypes. So scientists have realized that thinking of sex as a binary with only female and male and only XX and XY is really very simplistic and not reflective of reality. It turns out that science now says that sex is bimodal, meaning that most people fall into those definitions, phenotypically and genotypically, of female and male, but there are a significant number of people who are somewhere in between with either atypical sex phenotypes or atypical sex genotypes. So there's a great Scientific American article that maps out the spectrum of different intersex phenotypes and genotypes that I encourage you to check out for more information. But just know that this is the reality, that the reality is that sex is bimodal and there are a lot of intersex options. But for the purposes of teaching, it's often easier to simplify things, to first teach students about the rule versus then at later levels, more advanced levels, teaching them about the exceptions. So for the most part for the class, we'll stick with this simplistic definition, but know that it's not right. And it's just a model that we're going to use to be able to discuss the biology of the majority of the time. All right, so when gametes, our sperm and egg cells, form in the gonads, in the ovaries and testes, they are haploid. Remember, they only contain one copy of chromosomes and one copy of the sex chromosome as well. So since females have two X's, half of their eggs will contain one of the X's and half of their eggs will contain the other X chromosome. I tried to color them slightly differently to indicate that these two, half of their eggs will have one X and half will have the other X. And when it comes to males, half of their sperm will have an X and half of their sperm will have a Y. So when sperm and eggs combine in fertilization, there's a 50% chance that the offspring will be XX and female, and 50% chance that they will be XY or male. Note that the sex of the offspring is always going to be determined by the father. So depending on what sperm they get, all right, determines whether they are female or whether they are male. So it is the sperm donating parent that determines the sex of the offspring. Fun fact and digression here, chromosomal sex determination is not universal in the animal kingdom. Some animals don't, their sex is not determined by their chromosomes. It's determined by the incubation temperature of the eggs. And in some animal species, the adults can actually change sex at different points in their life depending on environmental factors. Some animals are hermaphroditic, meaning they have both. They're not intersex, they don't have some in-between male and female reproductive system. They have both a full male and female reproductive system. They're hermaphrodites. So there's a lot of variation in biological sex throughout the animal kingdom. Okay, so part of the reason that sex determination is more complicated than simply X and Y, male and female, is that the X and Y chromosomes, they are chromosomes. They are not single genes. And a chromosome is a really long piece of DNA. It's supercoiled into a bundle. So there's a lot of genes on a single chromosome. And we can map those genes onto different locations, or loci, which is the plural of locus, on the chromosome. Since humans are diploid and we contain pairs of chromosomes, so two homologous chromosomes, so each of our 23 sets or 22 sets have homologous pairs, it means that we have two copies of each gene. Those copies, though, aren't necessarily identical copies. They can be slightly different versions of the same gene. So I think of it as kind of like there are different flavors of the same food. So ice cream, there's strawberry, chocolate, vanilla. There's different flavors, but they're all ice cream. They're all the same food. So different genes have different flavors, and those different flavors of a gene are called alleles. So if you have two of the same chromosome, like I have pictured here, one from your egg donating parent and one from your sperm donating parent, all right, they might have the same alleles, which we would say are homozygous. They're same, homo meaning same, or they might be different alleles. They might be heterozygous. So in this example chromosome pair here, we're showing two loci, locus A, that encodes gene A, and locus B, that encodes gene B. All right, we would say that the alleles at locus A are homozygous because they are the same. They are both capital letter A. And the genes at locus B are heterozygous because one is capital and one is lowercase. So they are different alleles. All right, we have different alleles of the same gene here. So it is heterozygous. Now, the upper and lowercase letters do indicate something about these genes. It indicates whether the gene is dominant or recessive. It is dominant if it is capitalized, and it is recessive if it is lowercase. So these terms, dominant and recessive, came from the very earliest recorded studies of genetics by Gregor Mendel, who's often referred to as the father of genetics, and he studied the inheritance of different traits in pea plants. Now, this was before science even knew of the presence of DNA. We didn't have any of these terms. People recognized that children looked like their parents, that traits that parents had were often passed on to their offspring, and they didn't understand why or what the pattern was. And so Mendel did some very careful studies using pea plants to try to figure out mathematically what was going on and create a more defined explanation for inheritance. So in these studies with pea plants, one example of a study that he did was he noticed that peas could have different colors. They could either be green or they could be yellow. So there were sort of two different traits. And in order to study the inheritance of that trait, he took plants that were purely green. So when he crossed them with each other, they always had green peas, and ones that were purely yellow. So they always produced yellow pea plants. And he crossed them together, mom and dad here, and made a generation of offspring. And he noticed that all the offspring of the green plant and the purely green and purely yellow pea plants were all yellow. But then if he took two of those yellow pea plants and he crossed them, he got a generation that were 3/4 or 75% yellow, and about 25% of them were green. And he was able to explain this by saying that each of the parents must have a different have two alleles, right? Two versions of a gene, and they each pass one on to the offspring. And if it's a dominant trait, in this case the yellow is dominant, that if the offspring have even one of the dominant allele, they will express that trait. So only if they get two of the weaker or recessive trait, two copies of the recessive trait, will they express that trait. So in explanation, he coined these terms alleles and dominant and recessive and homozygous and heterozygous. So in this case, we have a homozygous recessive parent and a homozygous dominant parent, two capital B's. All of their children get one recessive and one dominant allele, and so they are all heterozygous. They all are capital B, lowercase b. So they all have a copy of the dominant and a copy of the recessive allele. That is their genotype. So their genotype is the alleles that they have, but their phenotypes are all the same because the dominant yellow trait is always going to dominate over that recessive green trait. So they all have the same phenotype, the same color. They express the same thing visually, right? But when we cross them and each parent has one dominant and one recessive gene, and they give them in different patterns, All right, the pattern that arises is that statistically, a quarter of these offspring will be homozygous dominant. They'll get a dominant B from each parent. 2/4 will be heterozygous. They'll get a dominant and recessive gene from each parent. And 1/4 will be homozygous recessive. And only those that are homozygous recessive actually are green. Do we see the green trait expressed? So this worked great with pea plants because, as it turns out, pea genetics are actually pretty simple. A lot of their traits are determined by one single gene, in this case the one I'm calling gene B, okay? Gene B leads to pea color. It's one gene for one trait. All right, but that's pretty simplistic, and human genetics are not usually that simple. So things in humans like eye color, hair color, height, these are controlled by multiple genes. So it's not just one gene that determines those things. And so the math on those types of traits is a lot more complicated and doesn't follow this simplistic Mendelian pattern. There are a few traits, though, that do follow this simple pattern. A lot of them are facial traits, so like freckles, if you have freckles, if you have dimples, if you have a cleft chin, those are all genes that those are all traits that are encoded by a single gene, so they do follow this simple inheritance pattern, and you can, determine inheritance patterns of those simple one gene traits in humans. Also, it's not useful for determining a lot of human traits since they're complex, but another thing that it is useful for is tracking genetic diseases, because genetic diseases usually do result from one single mutated gene. So we can use this pattern. So in order to study patterns of inheritance in humans, whether it's for traits like dimples or diseases like cystic fibrosis, it's very helpful to create what's called a pedigree, which is a sort of infographic diagram of a family tree that illustrates which family members have a trait, a phenotype, a condition, all right? In order to determine the pattern of inheritance and what type of gene we might be talking about. So just to kind of give you a tour of the key here, all of the circles in these pedigrees represent females, all of the squares represent males. Red is the color that's used to show whether someone has a trait and whether they have the dimples or have the disease, cystic fibrosis, etc. And blue is the normal phenotype, so they don't have that trait. And if it is half red and half blue, that represents individuals who are heterozygous. So they have the normal phenotype, but they do carry that recessive gene. And so they therefore might carry that condition or trait and could pass it on even though they don't have it or display that trait themselves, like those heterozygous yellow plants here in that generation. All right, so the first inheritance pattern we'll talk about is autosomal dominant. And in this case, if the parent has the condition, one of the parents has the condition, then there's a 50% chance that the child will get the condition. And that's kind of expressed here. And in this case too, you've got one parent has the condition, the other parent doesn't, and one of the kids has it, one of the kids does not, right? Notice that with autosomal conditions, autosomal dominant conditions, if you have the gene, you have the phenotype, all right? There are no carriers in this pattern because it's a dominant allele. So if you have it, whether you're homozygous or heterozygous, it doesn't matter, you will have the condition. So, yeah. in an autosomal recessive condition, all right, only individuals that have 2 copies of the recessive trait will have the condition. So in this case, if we're talking about cystic fibrosis, you have to be homozygous recessive in order to have the condition. All right, but lots of people can be carriers if they are heterozygous for the trait. So these 2 parents here do not have cystic fibrosis, but they both carry the gene for cystic fibrosis. And that results in a pattern similar to the P's on the previous slide, where about a quarter of the offspring will end up with cystic fibrosis, a quarter end up not, and about half end up with being carriers. Now, those statistics will happen if you had 100 kids. You'd probably see those numbers, but since each kid has a 25% chance of having the condition, that doesn't necessarily mean that 25% of your kids have the condition. It just means each individual kid has a 25% chance. You might have all kids with cystic fibrosis or all kids without cystic fibrosis. No, so that's a statistics problem right there. But the point I'm trying to make is that you can have offspring that are heterozygous, and those are individuals that are going to be carriers. They have the gene for the trait, but they don't have the trait, the disease, but they can give it to their offspring if they have kids with somebody else who is also heterozygous for the trait or homozygous for the trait. So notice here though that there's no difference so both males and females can be carriers. Males and females can both have the condition. There's no sort of sex discrimination with autosomal recessive conditions, unless they are sex-linked recessive conditions. So remember that so sex-linked recessive conditions refer to genes that are found on the X and Y chromosomes, really mostly the X chromosome, because the X chromosome is a lot bigger than the Y chromosome and has a lot more genes. So there's just very few conditions because there's very few genes that are on the Y chromosome. So remember, females, a typical female has 2 X chromosomes, and a typical male has 1 X and 1 Y chromosome. So if a female has let's say the disease that we're talking about here is hemophilia. So if there's 1 X chromosome that has the gene for hemophilia and 1 that is normal, all right, the normal one is dominant. The hemophilia gene is recessive, so the phenotype of this female will be normal and not diseased. When this female has children, all right, she'll give 1 X to each child. All right, the females will all get an X also from their dad, who is has the normal X phenotype. So the females will all end up either healthy or as carriers. But the males, depending on which X they get from their mom, could be healthy or could have the disease. And in this case, if they get the hemophilia X from their mom and they get a Y from their dad, then the only X they have is the one with hemophilia. So in sex-linked recessive disorders, they tend to have a much higher occurrence rate in males. Notice that all the red blocks on this pedigree are squares. That males are more likely to be affected because males only have the 1 X, whereas females get 2 X's, so they are more likely to be carriers than to be actually affected. All right? Females can have hemophilia. If the father has hemophilia and the mother is a carrier, they can make a child that has 2 X chromosomes with the hemophilia gene. And so you can have females that have hemophilia. Carrier females can also end up with hemophilia, though, because of a unique feature of X chromosomes called X inactivation. So none of the other chromosome pairs do this. The autosomal chromosomes 1 through 22, we have both homologous chromosomes are active in all of our cells. But it turns out that the X chromosome does have some genes on it that if you have both X chromosomes like active, you can sort of overproduce some genes that can lead to autoimmune conditions like lupus. And so evolutionarily, the body has sort of responded to this by inactivating one X chromosome in every cell if they are redundant. So which one is kept and which one is inactivated is very random. So what happens is when a zygote forms, when the gametes fuse and a zygote forms, there's two X chromosomes, and then the zygote divides, makes copies of itself, and embryonic development starts to happen. And at some early point in embryonic development, when there's a couple hundred cells or so, they all decide to simultaneously inactivate one of the X chromosomes in each cell. And which one they inactivate is totally random. So you get a mix, some cells that have inactivated the one from the sperm and some have inactivated the one from the egg. And then those cells go on to clone themselves further and further and differentiate into the different tissues of the body. And so a female is actually a mosaic of cells that some have one X activated and some have another X activated. And so even if you had identical twin females that had the same genomes and the same sets of chromosomes, they would have a different pattern of X inactivation because that pattern is very random. Now, in human females, there's very few genes on the X chromosome that produce any visible external traits, so you can't see a mosaic pattern externally on human females, but you can on cats. And that is because in calico cats, the gene for fur color is on the X chromosome, and there's an orange allele and a black allele. So you can have cats that are purely orange. They can be male or female because a male with an orange X or a female who's homozygous orange would be totally orange. If a female is homozygous black, and the male has a black X, then they would be totally black. But if you have a female that has an orange X or an orange allele and a black allele, then when the X becomes inactivated in the different cells, you get this mosaic pattern of orange and black fur due to this X inactivation in the cells, that only one of the alleles is expressed in each sort of cell cluster, resulting in this tortoiseshell or calico pattern. So in most instances, that calico cats or tortoiseshell pattern cats are always female, with a couple of exceptions, like the instance of a male that is XXY, that has two X's but also a Y. But for the most part, all of the tortoiseshell cats are going to be female, and it's due to this X inactivation that they get this mosaic fur coloration. So, okay, so far we've been discussing genetics at the level of chromosomes, but now we're going to zoom in and talk a little bit more about the structure of DNA, this double helix molecule, and the process of gene expression, or making proteins from that genetic code. So DNA, we know, is a double helix structure. It's a twisted ladder, if you will. It's made up of two strands of DNA. The backbone, or sort of sides of the ladder, are made of sugars and phosphates. This is what we call a sugar-phosphate backbone here. And the steps of the ladder are made by these nitrogenous bases that come in 4 different flavors, A, T, C, and G. So really the way that DNA is made is there are individual units called nucleotides that contain a single sugar, phosphate, and nitrogenous base, and then we link them together to make strands. These strands, you'll notice, another important feature of the DNA is that they run anti-parallel. So they're going in opposite directions. You can see that the letters of the bases here run down, and the letters on the other strand are going up. All right, they're going, this strands of DNA are going in opposite directions. Another thing about these about the DNA is that you'll notice that the base pairs only fit, the bases only fit with certain other bases. So A and T fit together, adenine and thymine, and G and C fit together, guanine and cytosine. So guanine can only pair with cytosine, it can't pair with adenine or thymine. We say that these are complementary bases. So easy mnemonic to remember those rules for base pairing is that A binds with T because the apple's in the tree, and C binds with G because the car goes in the garage. So the DNA encodes our genes, right? That gives us our genotype. The different genes in our genome reflect our genotype. But it is through producing proteins, through this process that we call gene expression, actually making proteins from the genes, that give rise to our phenotype. All right? And so this process in the cell looks like this. So the DNA gets transcribed in transcription into a molecule of RNA, single-stranded RNA, and that happens in the nucleus where the DNA lives. And then that RNA is sent out into the cell, either into the endoplasmic reticulum, which is not pictured here, or into the cytoplasm, where it comes into contact with ribosomes. And the ribosomes can read the RNA. This is a process called translation, and they turn it into an amino acid chain that then folds up into a protein. And it's these proteins that actually give rise to our traits, our physical traits that give rise to our phenotype. So our genotype does, in some way, determine our phenotype, but not necessarily. So that the thing you have genes for don't necessarily become a phenotype because the genes don't necessarily get made into proteins. Not all cells make the same proteins. And a lot of this gene expression is controlled by the environment of the cell. So there are different factors that control gene expression, and there's a whole field that studies this, and it's called epigenetics. Epigenetics, and it's very complicated, so we won't really get into it in this lecture, in this chapter, but it is really worth mentioning that epigenetics is the study of the control of gene expression. All right, how transcription and translation get turned on and turned off. And so how the same genotype could technically give rise to many different phenotypes, depending on when the genes are turned on and turned off. So epigenetics controls transcription. It's the study of the control of transcription and translation. So this is another way to sort of visualize transcription and translation. It's sometimes referred to, this process is sometimes referred to as the central dogma of molecular biology, that we have genes encoded in our DNA, and that DNA is then converted to RNA, really messenger RNA. That is a process called transcription, and the RNA is then converted to protein in a process called translation. It's, again, more complicated than this because of how transcription and translation can be turned on or turned off in order to selectively make different proteins. Okay, so let's talk about transcription. That's part one of gene expression, when the DNA goes to RNA. So in blue here, we have a double helix DNA strand. It's being unwound and opened up, by this enzyme here, the RNA polymerase, which actually it's another protein that does that, but the RNA polymerase is reading this code and transcribing it, making a copy in the form of RNA. So what is RNA? Well, RNA is very similar to DNA. They're like sister molecules. So they're both nucleic acids. That's what the Na stands for. So there's sort of three differences that we'll highlight between DNA and RNA. The first, most obvious one is that DNA is double-stranded and RNA is single-stranded. So I'm going to say DNA is double-stranded, RNA is single-stranded. Okay, that's the first difference. The second difference that's obvious is in that first letter, the D and the R. Well, they stand for something. So that second difference is the sugar in the nucleotide base. So if I go back real quick to this slide here, remember there's a sugar phosphate backbone. DNA uses a sugar called deoxyribose, and RNA uses a sugar called ribose. So they're slightly different sugar molecules. The third difference between DNA and RNA, which is kind of the most famous one, I think, is the difference between the fourth base. So they both use A, G, and C, but where DNA, the fourth base, is T-thymine, RNA's fourth base is uracil. Uracil is a very similar looking molecule to thymine, but slightly different. So it uses a different letter. One of the letters in the code is different, and helps us to distinguish whether a molecule is DNA or RNA. All right, so there's actually lots of different types of RNA molecules. RNA is actually really fascinating. The RNA that we are making in transcription is called specifically an mRNA molecule because it's a messenger. It carries the message of the gene code from the nucleus to the ribosomes, which will make our proteins. So what we are making in transcription is mRNA. All right. Then in the next step, translation, we're going to see that the mRNA interacts with rRNAs and tRNAs in the process of making proteins. So these are the three RNAs that get sort of the most attention in introbiology classes, but at higher levels, a really interesting type of RNA are these classes of regulatory RNAs. And these are studied a lot in epigenetics. A lot of these very small regulatory pieces of RNA play a role in turning transcription and translation on and off and determining which genes are expressed. So they are very fascinating. And primer RNAs are used in the process of DNA replication when we copy our DNA. And ribozymes are RNAs that can actually function as enzymes, which is usually a feature that only proteins can do. So anyway, back to this process of gene expression. So step one was transcription. Step two is translation. This is where we're going from RNA to protein. And I said there's different RNAs involved. So this long single-stranded nucleotide chain here is our mRNA. All right, this big blue machine here is the ribosome, and the ribosome it's an organelle technically. Well, not really, because it doesn't have a membrane around it, but it is a piece of machinery in the cell that's made up of protein and ribosomal RNA, rRNA, and it does the job of sort of reading this code and translating it into an amino acid code. So amino acids are the building blocks of proteins. And there's a third type of RNA, the tRNAs. These are transfer RNAs. They're the ones that bring the amino acids to the ribosome, and that's how this polypeptide chain is formed. So a polypeptide literally means many peptides, And these amino acids are held together by peptide bonds. Those bonds are called peptide bonds. So a polypeptide is really just another word for a protein. The chain gets all folded up. Once it's folded up, then we call it a protein. Okay, so the RNA polymerase is the enzyme that is responsible for transcription, for making the RNA, the messenger RNA, and then the ribosome is the enzyme that's important for making the, that's important for translation and making a protein. That's just the basics of gene expression. So we do need to go from DNA to RNA to protein to make proteins, but we also sometimes need to go from DNA to DNA. We need to make copies of our DNA when our cells are dividing. When cells divide, they copy their DNA, split it between the daughter cells. And that process is what we call semi-conservative. Oops. So semi-conservative means that when we make a new strand of DNA, we take the original strand, we separate the pieces, and then we make new pieces or new complementary strands. So the two new pieces of DNA, these two double helices, contain one strand from the original parent piece of DNA and one completely new strand. So it's not, we don't have one blue piece of DNA and one totally orange piece double helix here. So it's not like we have the old one and then we have a totally brand new double helix. It's that we have two double helixes and each of them has one old strand and one new strand. So that's what the term semi-conservative means. You're only conserving half. All right, so I also would be remiss if I just talked about the nuclear DNA, the chromosomes in the nucleus, because our cells do have more DNA than just what's in the nucleus, because mitochondria have their own DNA. But the mitochondrial DNA is very different than the nuclear DNA. It's not in chromosomes, it's in these little circular rings. And the mitochondrial DNA really only encodes genes that are important for the mitochondria to function. So very few genes there that lead to disease, but if they do, they lead to mitochondrial diseases. Now, the inheritance pattern, though, of mitochondrial DNA is different than the inheritance pattern for nuclear DNA, because when you have your gametes, the ovum, the egg, it's a relatively large cell. This is not drawn to scale. The sperm are very small. The ovum has a lot of mitochondria in it, and the sperm only has a few mitochondria. Those mitochondria are responsible for producing the energy that it takes to swim or move the flagella of the sperm. When the cells fuse, the sperm really just delivers the head portion, the nuclear portion of the sperm is what fuses and is delivered into the ovum, with the tail and all of the mitochondria that are in it kind of left behind. And so the sperm really only deposits its nuclear DNA into the ovum. So the zygote and all of the cells in the embryo contain mitochondria that came from the egg donating parent. And so mitochondrial DNA are strictly passed on through the maternal lineage, which is not necessarily, again, the source of many diseases, but it's particularly interesting for ancestry genetic studies because it can follow the maternal lineage, whereas sort of sociologically we pass on the name of the father. So you can usually through like marriage documents and stuff you can follow paternal lineage, but in using mitochondrial DNA you can follow maternal lineage. So it's kind of an interesting use. All right, so cells do need to replicate their DNA, but only when they are dividing. And when do our cells divide? Well, they definitely divide after fertilization. The zygote will divide to form the cells of the embryo, and those will divide. And really any process where the body is growing, we have cell division going on. But in a mature body, most of your cells are not dividing. They're just living their best lives, metabolizing, doing their jobs, like neurons and kidney cells. They're doing their thing, but they're not really multiplying. Some cells in the body do multiply regularly, like your skin cells. You're constantly growing new skin cells and old ones are sloughing off, so those are dividing more regularly. Anyway, when cells need to divide to make more copies of themselves, they go through a very regimented process of cell division that is called mitosis. So mitosis is a process of cells really cloning themselves. I'm saying themselves a lot. So you start with a parent cell. The first thing that happens is the cell will replicate its DNA, so it'll double all of its DNA, and then it'll split into two cells and evenly divide the DNA between them, so that these two daughter cells are diploid, just like the parent cell was. They contain two sets of chromosomes, and they are identical to the parent starting cell. So mitosis is a process of cells cloning themselves. And all the cells in our body will do this. Our somatic cells are basically all the cells in the body that are not our gametes. But remember, our gametes are not diploid, they are haploid. And so in order to make our gametes, they go through a similar but more complex division process that's called meiosis. And there's a lot of similarities. So in meiosis, you start with a parent cell, it replicates its DNA, okay? But in meiosis, the chromosomes, the duplicated chromosomes, go through this period where they swap DNA and they recombine. And that is called crossover. So the chromosomes literally like cross over and swap some DNA between the homologous pairs. And so then when the cells divide, they are technically diploid, but they're not identical to the parent cell. And they then go through a second division to split into haploid cells, all of which have slightly different combinations of DNA. It's hard to see, but this red chromosome has a little blue piece right here, and this blue chromosome has a little red piece right here, so they're kind of mixed and matched. So in mitosis, you end up with two daughter cells that are identical to each other and identical to the original parent cell. But in meiosis, you end up with four daughter cells that are haploid and have half the chromosomes of the original parent cell and are also not identical to each other. They're all slightly different. They all have slightly different combinations of genes. And so meiosis is only used, the only cells that do meiosis in the body are the gametes in this process of gametogenesis, making gametes, making eggs and making sperm. Now, this process of meiosis and mitosis, you can imagine how important they are to do them correctly, because DNA is the instructions for all the proteins you need in your cells. So you really need to do this process carefully, not make any mistakes, because if any mistakes are made, they get passed on to the next cells. And in the case of meiosis, if mistakes are made, those become the progenitor cells for a whole new organism, embryo. And this process is not foolproof. It is very tightly regulated, but mistakes do happen. And so mistakes in meiosis result in chromosomal mistakes in these eggs and sperm, but usually those mistakes aren't really seen. They don't display themselves until embryonic development. And so it's very common, something like 10 to 20% of pregnancies in the US end in miscarriage, usually in the first trimester. And most of the time, it's just due to faulty meiosis is to blame. Some chromosomal error that happened during haploid formation that didn't become evident until the embryo reached a point where it was becoming a problem and it couldn't develop any further. So there are some differences between gametogenesis in males and females that I want to highlight. So oogenesis is the formation of ova in the ovary, and spermatogenesis is the formation of sperm gametes in the testes. So some of the major differences. One is that oogenesis results in a single gamete, one ovum, and spermatogenesis results in 4 gametes, 4 sperm. And that's because in the process of oogenesis during meiosis, 3 of those 4 haploid cells get designated to die, essentially, and only 1 is designated to be the mature ovum. Versus spermatogenesis, all 4 haploid daughter cells are kept, are retained, and become sperm. So you get 4 times as many gametes during spermatogenesis than you do during oogenesis. So that's one thing. Another thing is the frequency of these events. So oogenesis occurs in the ovary once a month after puberty. Only one ovum is produced once a month. Versus spermatogenesis after puberty, it's happening constantly, continually, all the time. So every day, more and more of these sperm are being generated. So there's a lot more sperm generated than ovum. The third thing is that these primary oocytes and spermatocytes that then undergo meiosis to make our gametes are made differently in the ovaries and testes. So they both come from progenitor cells, and those progenitor cells are oogonia and spermatogonia. And they're sort of like a type of stem cell that can do mitosis and make lots more copies of themselves. So the oogonia can only do mitosis during embryonic development. So by the time a baby is born, their ovaries, the oogonia, have stopped multiplying. And so what's left in the ovary of a baby is just a bunch of primary oocytes that can't undergo meiosis until puberty. And so a lot of those primary oocytes actually die. They age and die before puberty even hits. And so there's sort of a limited number of primary oocytes in a female ovary that just age and get older with time. Versus in the testes, the spermatogonia continue to be able to clone themselves throughout the lifetime. So they can continue to do mitosis even after the embryonic phase, throughout the adult phase. And so new primary spermatocytes are being formed continually throughout life, which is why males retain their fertility because they're able to make sort of fresh spermatocytes throughout their lifetime, versus females are born with a limited number of primary oocytes and do not make more throughout their lifetime. So they can make ovum from that bank of primary oocytes, but the bank gets older and older, versus males always have sort of refreshed bank of spermatocytes to continue making sperm. So, in summation, in this chapter, we defined genome, chromosome, gene, allele, and other terms pertaining to the study of genetics. We talked about the different types of gametes and the process of fertilization on the chromosomal level, on the little bit of the cellular level. We talked about what genotype is versus phenotype. Genotype refers to the genes, the alleles that you have. You can be homozygous or heterozygous. Those traits can be dominant or recessive. And the dominance or recessiveness determines the phenotype, which trait is actually expressed, whether you have a disease or you have a external trait. We also talked about different patterns of inheritance using pedigrees to follow the transmission of a trait or disease to determine whether it is dominant, recessive, or sex-linked. We talked a bit about the DNA structure and how genes are expressed, how DNA becomes RNA, becomes protein. And we talked a little bit about DNA replication and cell division, both mitosis of somatic cells, the majority of the cells in your body, and that special type of cell division, meiosis, that happens in order to form gametes, eggs and sperm, our haploid cells. The end.